Hello, my name is Aleksandra Pfeifer. I work in National Research Institute of Oncology in Gliwice in Poland. Today I would like to talk about detection of germline exon copy number variants in targeted NGS panels for hereditary endocrine tumors and Lynch syndrome. We collected about 1,500 samples. All the samples were from germinal DNA. We prepared libraries using targeted enrichment methods and performed sequencing using mini-seq. Among our samples, there were 8 CNV positive cases according to former MPLA analysis. The aim of our study was to detect CNVs in our sample cohort using NGS and bioinformatic methods. We aligned reads using BWA-MEM and performed CNV detection using exon def using default settings. Additionally, we implemented our own tool for CNV visualization at single nucleotide resolution. Here, reads also were aligned using BWA-MEM. The calculation of def was performed with samtools def. Next, the normalization was performed using those two equations that were described in the paper of Schenkel. Finally, we obtained normalized defs, and we expected it would be equal about 0.5 in case of deletions, about 1 in case of normal copy number, and about 1.5 in case of amplification. Initially, we detected about 800 CNVs in our sample cohort using exon def, but it occurred that above 70% of detected CNVs were localized in the same gene, PMS2. We visually inspected those CNVs, and it occurred that there were probably false positives. It is known that there is a pseudogene, PMS2CL, that is highly similar to 6 exons of PMS2 gene, and therefore read mapping and CNV detection of PMS2 gene may be impaired. Therefore, we decided that instead of analyzing the def of PMS2 gene, we would analyze the sum of def of PMS2 and PMS2CL, and this solution was already described in the paper of Kerkhoff. After this enhancement, we detected above 200 CNVs in our sample cohort. It was on average 0.16 CNVs per sample. Here I would like to show a few samples which were analyzed both by MPLA and by NGS. Here we can see 5 samples that were first analyzed by MPLA. And in MPLA, the deletion of minimum 1 exon was detected in each of the samples. When NGS results were analyzed, also the same CNVs were detected. Here we can see another 3 samples that were first analyzed by MPLA. And in MPLA, the deletion of part of an exon was detected in each of the samples. However, in the NGS analysis, we did not detect those deletions. Here we can see another few samples which were first analyzed with NGS, and in all those samples, the deletion was detected. And then the MPLA validation was performed, and the validation result for all those 4 samples was positive. Here I would like to show the visualization of the deletion of MEN1 gene that was detected in one of our samples. On the upper left plot, we can see the visualization obtained with exon def. On the upper right plot, we can see the visualization obtained with MPLA. And on the bottom plot, we can see the visualization obtained with our own tool. And this visualization has a resolution of single nucleotide. Summarizing, we analyzed almost 1,500 samples and detected more than 200 CNVs. Our in-house visualization tool helped to visually inspect detected CNVs at single nucleotide level resolution. Combining ExonDev software with our own visualization tool may be useful in clinical and scientific practice. However, further validation experiments using MPLA are needed to evaluate CNV's detection performance in our dataset.